continuously worn away as the body ages and performs its functions. In the stomach, the acidic gastric juice digests the cells lining the wall of this organ and the upper part of the small intestine. About 500,000 of these cells are shed every minute. If this goes on all the time, how come you still have these organs of the digestive tract? Fortunately, the body has a mechanism by which the continuous loss of these cells in the stomach does not result in the destruction of the organ. This mechanism involves the formation of new cells that replace those that are destroyed. New cells are formed to replace the damaged or dead ones in many parts of the body. One way of reproducing these cells is called mitosis. The formation of new cells involves two processes. These are the division of the nucleus or karyokinesis and the division of the cytoplasm or cytokinesis. The process by which the nucleus divides to produce two new nuclei is called mitosis. This process may be divided into four stages: prophase, metaphase, anaphase, and telophase. Between mitotic divisions, the non-dividing nucleus of the cell is at interphase. At interphase, the cell is actively carrying out its many activities such as cellular respiration, protein synthesis, and growth. It is also during this period that the genetic material is replicated in preparation for mitosis. At interphase, the nucleus is prominent, but the chromosomes are not seen as distinct rod-like structures. Instead, they appear as stained granular masses or specks referred to as chromatin material. The nucleolus is quite visible inside the nucleus. In animal cells, a pair of centrioles is present in the cytoplasm near one side of the nucleus. Prophase. This stage prepares the nucleus for the separation of two complete sets of chromosomes into new nuclei. In an animal cell, at early prophase stage, the centrioles move to opposite sides of the nucleus and the chromosomes begin to appear as long, thin filaments. The nucleolus appears less distinct. Then the chromosomes begin to contract and appear thicker. The coiling of the filament into a tight helix possibly results in this highly contracted form of the chromosomes. The condensation of the chromosomes into thick rods is important to ensure that no fragments of the chromosome will be lost during distribution. Middle prophase, the centrioles move farther apart and start forming the spindle fibers. At this stage, each chromosome appears to consist of two strands called chromatids, which are held together at one point by a centromere. At late prophase, the nuclear membrane starts to disappear. The nucleolus is no longer visible. The chromosomes move toward the equatorial plate, attached by their centromeres to the spindle fibers. Metaphase. The chromosomes are arranged at the equatorial plate or middle region of the cell. The nuclear membrane disappears and the spindle soon appears. Each chromosome is attached by the centromere to the spindle fibers. The maximally condensed chromosomes align perpendicular to the metaphase plate. The chromatids in each chromosome separate from each other at the centromere. Anaphase. At early anaphase, the chromatids of a chromosome begin to move away from each other, one set going to one pole of the spindle and the other set going to the opposite pole. The centromeres become functionally double. The chromatids first separate at the centromere and the arms are dragged along. The poleward movement of the chromosomes is due to the contraction of the spindle fibers attached to the centromeres. At late anaphase, these two sets of chromosomes are farther apart and continue to move until they reach the poles of the spindle. Telophase. Each of the two sets of chromosomes regroup and become surrounded by a new nuclear membrane. The chromosomes begin to uncoil and now appear as long and thin filaments. The spindles disappear and the nucleoli gradually appear. Telophase ends when the new nuclei have the characteristics of interphase. Each of the chromosomes becomes so stretched that only relatively more coiled regions are observed as chromatin. Telophase completes nuclear division. Again, the four stages of mitosis are prophase, metaphase, anaphase, and telophase.